Again, thank you for the introduction, Simon. Yes, actually, we had a lot of conference in like pre-pandemic, but we're happy to be here again, and thanks for the invitation. We had some great talks today morning, and I know these are very much interesting. So the topic I'm going to talk today is on the biologics modality, is on the large molecules, the monoclonal antibodies. And just talking about my experience in the past, I was before at Novartis and Celgene, before BMS. And I used to work in the small molecules and all the melt extrusions and solid dispersions, all nanoparticles. So these are all my areas. And then about 4 years before, I actually went at BMS, take over the Celgene. I joined BMS, and that time I started working in some monoclonal antibodies. And that triggered some interest on me to work in this area. And from that time, I am now fully working on the biologics molecule. I'm still learning. So some challenging molecules, I'm the drug product lead, like antibody drug conjugates, the fusion proteins, the monoclonal antibodies, all these areas. So like before I moved to the biologics, we used to think that drug substance is the key focus on the biologics. But as I started working here, in my experience, the formulation, the technology, and going forward in the future, so these are some areas that are going to drive the biologic product development. And one of the key area, the challenging area, is the high concentration of biologic molecules or the formulation. So in that, I'm going to focus in today's discussion. So here, as the concentration or the dose is increasing in the biologics, because there are a lot of oncology molecules, but in the near future, we are getting a lot of molecules in the immunology space, and in that space, the dose is increasing, and also we have the patients. So as in the morning, there were some discussions about the patient compliance, the patient's comfort, right? So these are the areas that we need to focus now, and for that, we need to have a better formulation, and we have the dose increased, so we cannot just keep the IV in the hospitals. So we have to make it like a house administration to make it more patient-friendly, or once in a month administration, because our competitors are like the small molecules, like oral tablets. So obviously, there are a lot of challenges in this area, and in my slides today, I'm going to focus in this. So we recently actually got a paper published, and you guys can refer it afterward, but I'm going to cover about some of the formulation aspects and other areas from the commercially launched some of the molecules. So far about 120 of the antibodies has been launched in US market. And of the 120 antibodies, there are about 50 antibodies that are high concentration. And so when I talk about high concentration, this is about 100 milligram per ml or more. So we have taken all the molecules that are launched, and we take the key identifiers, and mainly the formulation aspects, all the technology, the devices that are used, and we assess that so that we have a good database for the future development, and also to answer some of the questions coming from the agencies. So this is one of the molecules. I'm just taking an example. The identifiers we have used. This is from BMS Orencia. This is a very familiar it's a molecule which was launched in 2005. So basically, we have assessed the antibody, the type of the antibodies, then the concentrations, the route of administration, the packaging, the dosage forms, and many of the formulations. And that's what I'm going to have some more detailed discussion today. The containers, the pH, and all other aspects. So just going back, so this is the initial approval that happened of all the antibody molecules. So as I told before, so 120 antibody molecules were commercialized in US. And on that, this graph is showing about 46 molecules that are launched with a high concentration, which means more than 100 milligram per ml. And as you see here, in the right-hand side, there are a few blockbuster compounds from the small molecules, from the fusion proteins, from the antibodies, like Humira, Keytruda, Orencia. So these are some of the familiar compounds in the biologics. So these are all blockbusters. So obviously, there is a huge market. There are about 4,000 molecules in the antibodies that are in clinical studies in different phases, from phase 1 to phase 3. In 2018, the market is about 115 billion, is increasing. In 2019, about 150 billion, and we're expecting by 2025, it will be about 3 billion or more. So obviously, there is a huge pipeline. Even at BMS, we have more than 50% of the molecules in the pipelines are the biologic programs. There are more programs coming in the ADC in all industries. So there is a huge potential for innovation in the formulation side. In this slide, I'm showing some of the therapeutic areas. So obviously, oncology and respiratory, cardiovascular, neurology, these are the different areas in which all the biologic molecules of high concentration has been launched. But one of the area in blue here is the immunology. So that is one of the key area that is driving some of the challenges and some of the developments in the biologics within the space of the drug product development. In the right-hand side, I'm showing some of the concentration. So by definition, it's not like that if it is more than 100 milligram per ml is a high concentration, but we have just taken the number because we have seen once it goes above 100 milligram per ml, there is a lot of challenges on the viscosities, on other stability issues. So we have taken all the molecules that are launched from 100 milligram per ml or more. As you see here, from 100 to 125 or 24 products. And then we have 17. Even there are few products that are launched with 200 milligram per ml. So these high concentration products are increasing. So what's the basic challenge here? So in the next slide, I'm going to discuss about the different route of administration. But one of the very familiar route of administration is the subcutaneous injection. In the subcutaneous injection, it is limited to 2 ml. So we can administer, as you know, about 2 ml of the volume of injection. And that's the challenge. So as the dose is increasing, as the concentration is increasing, it is getting more and more challenge to administer these products. And that's why we need more development. So as we have done the assessment, there are a few products that are in IV, and these are mostly the oncologies on other areas. Between 25 to 100 milligram per ml, there are a lot. But in the area of the high concentration, we have mostly the subcutaneous. And obviously, there is a reason for that, because in this space of the immunology space, because when the patients are taking the dose, not going to the hospital, right, because they want to take in home or from a clinic. So for that, they need a more patient-friendly administration of the dose, and that's what the subcutaneous are getting more and more familiar. So let me discuss about some of the chemistry part before I jump into all the attributes. So this is a typical cartoon of the antibody. As you see here, the blue portion is the Fc region. So this is the Fc, the constant. This is the antibody, which is the immunoglobulin part. And the pink is the variable region. So basically, the immunoglobulin part, which is the blue region, is more or less constant between these molecules. This is the immunoglobulin part. And therefore, we are lucky that we don't have to do like more variability in our formulation development. So but the variable region, which is the pink part, that is more challenging or more important, because when we do the development. In this variable region, we have this domain part, which is the antigen, which links with the antibody. So that is, again, like very much important. So when we do the development, we generally do some mapping at the very early phase of the development to understand what are the key domains. So there are about 120 amino acids in the whole antibody. But we have to see what are the domains that are sensitive to some of the physical or the chemical degradation. And we see the part which are more linked with the CDI region, the region that get attached with the antibody. Because if there is some change in the protein in this CDI region, so that will impact the interaction with the antibody. That will impact the binding. So that will impact the bioavailability. So, and also the clinical outcome. So therefore, it is very important to map and understand what are the sensitive area in the variable region that get attached with the antibody, and that's what our focus will be when we do the formulation development. So as we're discussing with the dosage form, so let me first with the dosage form. What we have seen in this high concentration, as you see here, the most of them, about 80 to 90% of the products are launched as the ready-to-use, the liquid formulation. There are few products in the lyo formulation. So obviously, the lyo process, has some limitations. Like this is a more complicated and this is more expensive process. And not only that, it can also have some negative consequence on the protein stability. So if it is not needed, we generally don't do the lyo. But obviously, if we need in the stability, if this protein is not stable in the liquid form, like some of the ADC compounds, then we could just easily go with the liquid formulation. So most of the products in BMS and also, as you have seen, in all the commercial antibodies are launched as a liquid formulation. So this formulation generally comes because when you get the drug substance, we get the frozen drug substance, and we take the frozen drug substance and fill in the vials. So this is as one approach. And so the primary packaging, as you see here, as we're discussing about the vials, so far many of this product has been launched as in the vials, as a packaging, as a primary container. But this is the trend until 2015 or 16. But in the recent years, it is getting more and more focus on the devices. Because as we are getting more into the area of the immunology, like home administration, the more patient-friendly, so the devices are getting more focused. And in terms of device, I mean the pre-filled syringe or the auto-injectors. So if you see one of the graph in the right-hand side, the pre-filled syringe, there is obviously an increasing trend in the pre-filled syringes because these auto-injectors and the pre-filled syringe are more safe. We don't need to give some overage and are more patient-friendly. Even the auto-injectors are getting more popular. There is only few products in the market which are having the auto-injector, but in the next few years, there will be more and more coming. So the auto-injectors are, again, more patient-friendly in terms of the pain and patient comfort. And again, we have few products in the market, and one from BMS is our 2 ml auto-injector. But still, we are limited to the volume. We need products. There is no products in the market with a large volume auto-injector and where there are a lot of innovations are going on. And similarly, the slow infusion pump, I will discuss later about the device. So these are some of the areas which will be getting more and more popular in the near future to administer a high concentration or high volume. So let me discuss about the risk of the formulation. So in general, a 50 milligram to 100 milligram per ml generally is more stable. But as we increase the drug concentration more than 100 milligram per ml, then we see there are challenges on the stability. And there is more aggregation because the proteins are getting more and more crowded. And there is aggregation, there is increase of the viscosity, and this impacts the overall stability of the product. This impacts the process in terms of the filling. There is more shear stress, interfacial stress. So these are some challenges as we increase the concentrations of the protein, and that's what we need to optimize the formulation in order to stabilize the formulation. So first, let me start with tonicity agent. Once we have taken all the products from the commercial, about the 50 products that are with a high concentration between 100 to 200 milligram per ml, we have seen that sucrose is most frequently used as a tonicity agent. So sucrose is a very good stabilizer, which stabilizes the conformational stability of the proteins. And sucrose is used both in the lyo and also in the solution. One of the key advantages for sucrose, it helps to remain the drug in the amorphous form during the freezing, during the lyophilization. And also because when we freeze the drug substance. So this is one advantage for the sucrose as compared to the other components like trehalose, sucrose, and mannitol, because this crystallizes during the protein. So obviously the trehalose, the sorbitol, the mannitol has been used in the product, but we have to take additional precautions when you're using like the trehalose, sorbitol, or mannitol in the formulation. The buffer and the formulation pH is one of the most important, the key driver of the formulation. So in order to stabilize the formulation, we need to optimize the buffering agent and the concentration and also the pH. So different buffers has been used in all the commercial products, but one of the most familiar is the histidine, and also in combination with other buffers. We sometimes use a combination of buffers. I think the key objective is about the charge variant. So we have to optimize the charge in like in the antibodies. So this is very much important. So that is optimized by optimizing the concentration of the buffer and the pH. So in general, the pH range from 5 to 7, but this pH actually controls some of the degradations as we're discussing before. Like at the low pH, we see more isomerations. At the high pH, we see more deamidations. Just an example. So it is very important to do a pH profile study at the very early phase of the development and then to optimize the pH. And we have seen this is so much sensitive to the pH because if there is a small change of pH or the buffering capacity, it can disrupt the whole protein structure and destabilizes the protein. So it is very important to understand the scientific aspects of, because when you do the mapping, it's very important to understand the mechanism of the degradation, and from that, how to stabilize this antibody by controlling this formulation pH and also the buffer. So of course, so most of the antibodies that are being launched have the buffer, but we see one of the antibody is a very familiar compound from AbbVie, which is the Humira. So Humira is first launched with a citrus buffer, but then we have some problems from the market. So this is a pain. So it is very well known in the biological field. We do immunological studies. We do the pain study. So citrus buffer is one of the buffer that sometimes alleviates the pain in the patients. So later, this product was launched as a buffer-free, as a citrus-free product. And it was seen that the pain has been eliminated, the patient comfort has been increased. So this formulation that has been launched has no buffer, although the concentration was about 100 milligrams per ml. So this is also a very important fact that these antibodies also sometimes acts as a own buffer in the high concentration. So which is another important thing that we need to consider. The surfactants are also very much important in this area because surfactants, these antibodies are very sensitive to all the stress or the interfacial stress that is going on during the transport, during the mixing, even during the handling. So surfactants are very much important to stabilize these molecules by lowering this interfacial stress. Because we have seen the surfactants like polysorbate 20, 80, these are most commonly used in these antibodies. There is also one more important factor because when we do the protein purifications in the upstream process, sometimes we have the lipase enzyme. So this enzyme we try to control is very important to control this enzyme, but this lipase enzyme is also sometimes results to the decrease of the surfactants because it breaks down the surfactants, the polysorbate. So this is the requirement from the agency that during the stability, we need to assess the surfactant concentration in the formulation. So again, surfactant concentrations are one of the key formulation parameter and need to be optimized in order to stabilize the formulation. So as we are talking about the high concentration, so viscosity is a very important parameter that we need to address. As the concentration is going up, then 100 milligram per ml, there is also a linear or exponential increase of viscosity. And therefore, this viscosity reducing agent need to be considered. As we have seen the product like some of the amino acids and the salts, the hydrochloric salts of the amino acid like arginine, sodium chloride, proline, glycine. These are some of the key components that are used to reduce the viscosity of the proteins. And there is another innovation. So in this area, there are a lot of innovations are going on. A lot of companies and a lot of excipient companies are coming up with some of these innovative excipients that can be used to decrease the viscosity without impacting the stability of the molecules. So in summary, as we have seen, so these are some of the key excipients. We talk about the buffer to maintain the stability, to maintain the pH and stabilize the molecule. Histidine, phosphate, acetate, these are some of the key buffer. We discussed about the sugar, which is the sucrose, in addition to the trehalose, mannitol. And then we discussed about the tonicity. Again, the sucrose has been used. We also discussed about the surfactants, the polysorbate 20, 80. In one or two products, we have also used the polysomer 188. Then we also discussed about the viscosity modifiers. The two other additional excipients is the chelating agents. Yeah, because we have to also prevent some of the metal-induced degradation. DTPA and EDTA, these are most commonly used in some of the formulations. And last but not the least, antioxidant. So methionine is also used antioxidant, but in very rare cases. To switch gear now, let me talk about the bioavailability. So like today morning, we're discussing about a lot of the bioavailability of the small molecules. We heard about the BCS classification. In this area, this concept, we are still investigating on this, and there is one good paper we have discussed about a very similar of the classification like BCS, like 1, 2, 3, 4. So the key attributes when we inject the protein solution in the hypodermis, the two key areas that we need to focus is the molecular transport of the molecule under the skin and also the extent of the catabolism. Like as we're discussing in the small molecules, like the solubility, right, or dissolution. So as here we have the molecular transport and the catabolism. So if the catabolism is low, the molecular transport is high. Obviously, this is a BCS. It is like a BCS class. This is a class 1. And if the catabolism is very high and the molecular transport is low, this is becoming a class 4 compound. So this is how we are trying to categorize these or the biologic proteins. And if we are able to address these two important parameters, then we will be able to enhance the bioavailability. So an application of this concept we see in one of the technology that is being widely used in the last few years. This is the enhanced technology by using the hyaluronidase enzyme. So what it does, the enhanced technology uses the proprietary the human hyaluronidase as the enzyme to enhance the transport or to enhance the bioavailability of some of the proteins. So it's basically not directly linked to enhancing or increasing the bioavailability of the proteins. So let's see how it works. So in the cartoon, if you see the left-hand side, so in the hypodermis, there is a hyaluron. This is the hyaluronin enzyme, which is like a very network, is a rigid structure. So when the high concentration of the proteins are administered by the subcutaneous, so there is obviously some limitation of the transport. The transport is not great. There is also catabolism. So obviously the bioavailability, there is some variability. The bioavailability is not great. But when we administer this enzyme, it helps to disrupt the structure. So this actually helps to depolymerize this enzyme, increase the transport mechanism, and decrease the catabolism. And this is how this technology is helping to enhance the bioavailability, to reduce the variability of some of the high concentration proteins. And also we can administer in large volumes. So there are a few products already in the market. As we see here, most of them are the fixed dose combination. As we see here, the concentration is about 120 milligram per ml for all the products, interestingly. But it's the dose volume that we are able to increase. As I have discussed before, the 2 ml is a cutoff point, right, for subcutaneous. But by using this technology, we are able to go up to 5 ml and also up to 15 ml, 10, 15 ml. So this technology obviously helped to enhance the bioavailability. So in conclusion, I have discussed about the RTU solution, about the histidine, about the buffer-free, about the enhanced technology. So I have a few more slides that I will discuss about the future trend. So as we see here in this graph, so we have the viscosity, number 1. Number 2 is the dose volume. The viscosity is again related to the concentration. So the concentrations of the viscosity and the dose volume, that are the area that we need to focus in the next few years in biologics. So obviously this graph is discussing about 10 CPS or less viscosity, or about 2 ml or less, like 3 ml or less. So those are the products already being commercialized. We have used the enhanced technology, which is the green area, to increase the volume, right? Although this paper has talked about the 10 CPS, but based on our experience, we can still go up to 20 CPS. So as long as we stay below 20 CPS, I think we are fine. The volume is about 2 ml. But once we are going to with the viscosity of more than 20 CPS, and the volume more than 2 ml. That's what we need, the technology, the more enhanced technology in formulation and devices. So I'm not going to go in the details, but here in the next 3 slides, I have given some of the examples of the innovative work that is happening in terms of the formulation. So this slide is talking about the formulation, and again, these are all the experience we are getting from the oral solids, right? So by using a particle engineering process, by changing the parameters of the particle or the attributes, by using like a dispersion technology, by using conjugation technology, we are able to address some of the challenges on the viscosity. Like the Zeris technology and the electrified technology, these are some of the key technologies that, that many industries are collaborating and start working. And similarly, in these 2 slides, I have discussed about some of the devices. So these devices are also getting popular and it's neat. So I'm working on like compound in BMS. So we are in the phase 1, like we haven't actually started the SAD study, but I have to plan even the commercial strategy because these devices are not only expensive, these are $1 million to develop, these also take a lot of time. So in the commercial, we have a 2 ml device, but we don't have a large volume auto-injector. So that's what, there are a lot of developments that are going on this area. And similarly, the infusion pump and wearable injectors. So these are the slow infusion pump where the patient can put this device on the body, this on-body device with a slow infusion, and by which we can be able to give a large volume or a high dose. So these are again some of the areas that we need to focus in the near future. And last but not the least, the fixed dose combination, like again oral solids, these are also getting very popular. So far we used to do the co-administration, but now we need the co-formulation, right? Again, to make it to make the formulation or to make the product more patient-friendly, more life cycle management approach, we need the co-formulation, which means again we have to administer in a high dose and a high concentration and high volume. So in conclusion, and the future, obviously, this is the area in biologics, these high concentrations or high volume, we need to focus, a lot of innovations are going on. This is the area that we need a lot of technologies, a lot of optimizations in the formulations, a lot of challenges. And as I have discussed, there are a lot of opportunities on the formulations, technologies, and the device that we need to improve the dosage form that will be more patient-friendly, that can compete with the other products in the market. So thank you. I think there's some time for questions.
Right. Thank you, Indrajit. Yeah, we've got a minute. So any quick questions at all on antibody ABCs? Yes, got a couple of.
Thank you, Indrajit. Nice presentation. I was wondering about how the concentration of the antibody, increasing the concentration may affect pain for the patient, injections and reactions, and if you have any information about how that affects bioavailability as well.
Yes, so that is one of the areas that we are focusing also, and that's a very important question. So once we increase the concentrations or the volume, this also impacts some of the bioavailability, right? And that's what we were discussing, because once we're injecting as a subcutaneous, this all the catabolism and the transporter mechanism, so this can actually impact your overall PK, and that's why when we increase the concentrations, we do all the PK studies, we do some modeling work, we do the PK studies in the mini-pig, and before we go to the human. So obviously we study that area, but at the same time as we're discussing the slow infusion pump, the slow infusion is also very important in that area to control some of these challenges in the PK aspect. Now, as we see here, all the products that we have commercialized so far, the bioavailability is not so bad as like a solids, as the oral. So it is like from 60% to 80%, but there can be a lot of variability. But even if we increase the concentrations, it can impact, right, the BS. So that's what we need to focus a lot on the biopharmaceuticals aspect in that area as we increase the concentrations of the volume. So that's a good question. Thank you.
Any last questions? Oh, yes. One more.
Well, thank you for the presentation. It was very interesting, and it's nice to see that some of the challenges that we are facing are very similar. So I just wanted to know if, in terms of the co-formulations, with regards to the PK, is there any challenges that you have, or any, differences with respect to individual PK, individual agent PK, and once they were put in a co-formulation product, if you've,
Yeah, again, a
faced
good
any of
question
those
on,
challenges?
right. Yeah, we have, again, I'm not the expert on the biologics. I started working like in 3 years, okay, so I'm still learning. But I know, as we're discussing, because when you co-formulate the product, these are the defined PKs. Now, antibodies, again, are not as worse as the oral, right? It's not like a 5% or 10%, 50%. It's still within the margin, but still we do the PK aspects of the individual molecules to see if any impact on the PK. I don't see any literatures, again, from the co-formulations that there is a drastic impact on the PK. But yes, because these are some of the life cycle management approach. So once we are moving from a mono compound to a fixed dose compound, we need to understand the PK aspect from each animals and then human, okay? So, and we also do relative BS study before we switch into that. So that is one of the key area that we focus in the next future, all the fixed dose compounds, all the formulation aspects also. And the last thing is also, I think it is very important, a platform formulation, right? So it's better to have a platform formulations in organization. I know this is very complicated, again, for the oral solids. In the biologics is more well behaved, like the antibodies, within a certain concentration. So if you have the platform formulation, I think the impact of variability of the excipients, can be minimized. So once you combine two different formulations of two molecules. But obviously, it can impact the PK that we assess. I hope I would answer your question. Thank you.
Okay, let's give Indrajit a round of applause again. Thank you.
Thank you.